University of Maryland, College Park. So today I'm going to talk about this new line of research from our lab called Cuttlefish. The manuscript is titled as Cuttlefish, Fast Parallel and Low Memory Compaction of de Bruijn Graphs from Large-Scale Genome Collections. So to give some context into the work, with the increasing throughput of sequencing, today we have thousands of mammalian genomes. We also have genomes that are much larger to typical mammalian genomes. For example, we have the sugar pine genome, the Mexican walking fish, or the axolotl genome. These genomes are larger than 30 gigabase pairs in length, compared to a typical mammalian human genome of like 3 gigabase pairs. So with the increasing affordability of these whole genome references, there is an increasing need for efficient sequence indices to align long and short reads to these references. There's also a need for basis data structures that we can use for comparative genomics and pangenomic analysis. Hence, compact representation of these reference collections is of acute interest to us. So the simplest form to represent a set of references is to have like each reference as just a plain string. For example, here we have two strings from two different references, a blue reference and the red reference. The green blocks here depict identical substrings. For example, these two green blocks here are two repeated substrings across these two different genomes. Similarly, these three purple blocks represent the same identical substring that's been repeated 3 times across these two references. So instead of just these plain strings, what we may intend to have is something like this, that is where the repeated substrings or repeated subblocks of nucleotides should be unified into one block. For example, we may want to have a graph structure that looks like this, where the repeats are resolved into one block. So an excellent object to represent these reference collections and sequence reads is de Bruijn graphs. So historically in bioinformatics, de Bruijn graphs have been extensively used in genome assembly algorithms, and in recent times it's been increasingly used in sequence indexing stuff and comparative genomics, pangenome analysis, and so on. And for these applications, we are usually interested in the colored compacted variant of de Bruijn graphs. So to give a very brief refresher on de Bruijn graphs, consider these two references, this red reference here and this blue reference here. The graph at the top part, this one, this is a de Bruijn graph for this red reference using k equals 2, that is we are using tumors as the nodes, and the graph at the bottom is a de Bruijn graph for the blue reference. Now if we want to represent the de Bruijn graphs in a combined manner, that is take a union de Bruijn graph of these two references, then the union de Bruijn graph is this graph at the bottom. That is, this de Bruijn graph is a colored de Bruijn graph that represents the red reference and the blue reference simultaneously. So introducing de Bruijn graphs in whole genome analysis pipelines highlights a set of different algorithmic challenges. For example, how do you construct this de Bruijn graph? How do you efficiently represent de Bruijn graphs in memory? How do you build efficient indices on these graphs? How do you align reads using these indices to the graph? So for these pipelines, in this work, we intend to tackle the initial step, that is, how do you build the colored compacted de Bruijn graph in a time and memory efficient manner? So we use bidirected de Bruijn graphs in the work. So we are not going into too much details into the preliminary definitions and stuff, but in a bidirected de Bruijn graph, each canonical k-mer corresponds to a vertex of the graph, and we say that each vertex has two sides, the front side and the back side. So in this picture, for each vertex, for example, this GAC vertex here, The flat side, the vertically flat side is the front side, and the vertically pointy side is the back side. So the sites information is important when you are visiting a canonical, when you're visiting a vertex. When you're visiting a vertex, if you see the corresponding k-mer in the canonical representation, then we say that we are visiting the vertex from the front side. Otherwise, if we see the k-mer in its reverse complement form, then we say that we are visiting the vertex from the back side. And what are compacted De Bruijn graphs? When you for the original De Bruijn graph, when you collapse each of the maximal non-branching paths into a single vertex, then you get the compacted variant of the graph. For example, for this for this De Bruijn graph here on the left, there are 4 maximal non-branching paths: this one, this one, this one, and this one. Collapsing each of these branches, maximal non-branching paths, into a single vertex gives us this graph at the right. So this is the compacted version of this De Bruijn graph. So in this work, we try to build this compacted De Bruijn graph given a set of references. Given enough computational resources, the problem is not that hard to solve. That is, given a set of references R, a naive compaction algorithm may go like this. First, you build the ordinary De Bruijn graph by going over each of the input references. Then, using some linear time graph traversal algorithm like the DFS or the BFS or variants of it, you can, and using some other bookkeeping, you can build the compacted De Bruijn graph easily, given that you have the original De Bruijn graph at hand. Now, this naive algorithm is infeasible for moderate to larger sized genomes because the space requirements for the ordinary De Bruijn graph is prohibitive. So, so we need to somehow bypass the construction of the ordinary De Bruijn graph and build the compacted De Bruijn graph directly given the set of references. So, a motivation, a fact that we should remember, note as important, that even without having the ordinary De Bruijn graph at hand, we can make an implicit walk over the graph. In an edge-centric De Bruijn graph, each k+1 mer from your input collection corresponds to an edge in the De Bruijn graph. So if we go over, if we scan over the input strings, we are encountering each k+1 mer one by one. So we are visiting each edge one by one, and the edges being adjacent. So in this manner, we can have an implicit walk over the original De Bruijn graph without having the graph explicitly, in some explicit format. So this gives us a way to walk over the graph without having it. So, and all the maximal unitics, we haven't defined unitics in the earlier slide. The maximal non-branching paths are referred to as maximal unitics. All the maximal unitics are contained as subpaths in these walk traversals. So we need to somehow extract off these subpaths from these implicit walks. In this work, the concept of flanking vertices is quite important. So we say that if we can characterize a vertex, each vertex of the actual De Bruijn graph as either flanking or internal with respect to the maximal unitics, then if we can characterize the vertices somehow without extracting the unitics first, if we can characterize the vertices as flanking or internal, then we can make an implicit walk over the graph again and extract all the subpaths that have flanking vertices as its terminal ends. That is, if a subpath is started with a flanking vertex and ends with a flanking vertex, then it corresponds to a maximal unitig. So this observation reduces the problem of graph compaction to the problem of determining the set of flanking vertices. So to formally characterize flanking vertices, a vertex V in the De Bruijn graph is referred to as a flanking vertex if this vertex has some side as V such that that side has either zero incident edges, that is no incident edges, or more than one incident edges, Otherwise, this side has exactly one incident edge, but that edge connects that side to some other side of another vertex, which side, I mean that another side has multiple incident edges. Let us see an example here. I mean, this vertex here, AGA, at my mouse pointer, this vertex has multiple incident edges at its backside, so it satisfies the criteria 1, so that's why it is a flanking vertex. And this vertex CTC here, it has unique incident edges at each of its sides, but the edge at its backside connects it to this AGA, this backside of AGA, which has multiple incident edges. That's why CTC is also a flanking vertex. So that's how we characterize the flanking vertices. So we can say that each flanking vertex can be in, each side of a vertex can be in 5 different configurations. Suppose that the side of a vertex has a unique incident edge, then what nucleotide, what's the nucleotide, based on the nucleotide that encodes this edge, there can be 4. So based on which edge, which unique edge it is, there can be 4 different configurations. Otherwise, this side has multiple incident edges, and there can be a bunch of scenarios like that. We group all these scenarios into one configuration. So in this way, we say that a side of a vertex can be in 5 different configurations. 4 configurations for each unique possible edge, and 1 configuration for all the rest of the cases. In this way, we say that a vertex can be in 25 different configurations, since a vertex has 2 sides and each can be in 5 sides. So, and we call each of these configurations as states. So this provides us with an idea that we can treat each vertex as a deterministic finite state automaton, or DFA. So a DFA is formally defined using a 5-tuple. Due to time restrictions, we are not going into all the details of all the parameters, just a few. Q0 is the initial state of the DFA of each vertex, which is the unvisited state. Before making any graph traversal, we do not know any information about any of the sides of any of the vertices. So each vertex starts from this unvisited state. QPrime is the set of all possible 25 different configurations of states. Now, based on the structural properties of each state, the 25 states can be characterized into 4 disjoint classes. For example, if you see the first shape here, that is this state, which has a unique incident edge at the front encoded by x1 and a unique edge at the back encoded by x2, there can be 16 different configurations or states of this sort. So all those 16 states fall into this single in, single out class. Similarly, there are 3 other classes like multi-in, single out, single in, multi-out, and so on. And lastly, delta is the transition function. This is the most important parameter in this DFA, and delta governs how each of the automata transitions from one state to another. This picture, this is a very detailed overview of how the transition function works. So let's not dive too much into it, but let us just simulate the transitions for a random hypothetical k-mer that it may encounter during our graph traversal. So each automaton, that is each vertex, starts from this unvisited state Q0. Say that for this vertex, the first time we see this vertex in our walk, we see that the front edge is encoded with a C and the back edge is encoded with a G. In that case, this vertex, or the automaton of this vertex, is transitioned from the state Q0 to this state which belongs to the single in, single out class. This state is one of those 16 states from that class. So this vertex remains in this state as long as for all the next occurrences of this vertex, we keep seeing the C encoded edge at the front and the G encoded edge at the back. Now say that at some occurrence of this vertex later, we see a different edge that's not encoded with a C, but something else like A, T, or G. But the back edge is still G. In that case, that vertex is, this automaton is transitioned from this state to this state of class multi-in single out. Now from this point onwards, we do not care about the edges that are incident to the front side, because these are not relevant for our purposes anymore. And as long as we keep seeing the G encoded edge at the back, this vertex or this automaton remains in this state. And if later we see something else other than G, in that case, this automaton will transition to this last dead end state, which is multi-in, multi-out. So that's one hypothetical scenario that one K-mer might follow during our graph traversal. So this is a very high-level overview of the possible transitions between the state classes, between the 4 state classes. Since we are associating, we need some associative data structure that associates the state of each vertex or each canonical K-mer to the K-mer. So for that, we use a custom hash table designed. For this hash function, that is to associate the state information to each canonical K-mer, we use a minimal perfect hash function as the hash function using the BB hash algorithm, which is a very efficient MPH, minimal perfect hash algorithm. This is possible because the set of keys, which is the set of canonical K-mers, this can be enumerated before we go on making the graph traversal. Now, representing this hash function takes around 3.7 bits per K-mer, irrespective of the K-mer size. To store the states information into the hash buckets, we use a bit-packed array. Now, since each vertex can be in one of 26 states, that is 25 states plus the unvisited state, since there can be 26 different states, we need at least 5 bits per K-mer to succinctly represent the states. So as a whole, our hash function takes 8.7 bits per K-mer, irrespective of the K-mer size. So the whole algorithm, as to give an overview, has 4 steps. At the first step, we extract all the unique K-mers using the KMC3 K-mer counter algorithm. Then, once we have the set of K-mers, we build a minimal perfect hash function using the BB hash algorithm. Then, after we have the hash table structure, we make a graph traversal, and using the DFA modeling scheme, we compute the states of each of the vertices. And then using another graph traversal, we extract off all the maximal unities by characterizing the flanking vertices on the fly. So to give the asymptotics, if our set of references has a total length of m and there are n distinct K-mers, then the running time is loosely bounded by this m plus n times k over 32 plus h, where h is an expected constant, and the memory usage is 8.7 times n, where n is the number of distinct K-mers. So we are using roughly a byte per distinct K-mer. To assess Cuttlefish against state-of-the-art algorithms for compacted de Bruijn graph construction from reference sets, we use 2 different kinds of experiments, one for individual genome reference compaction and one for colored de Bruijn graph compaction. For individual compaction, we use 3 datasets: human dataset, western gorilla, and sugar pine. And for the colored compactions, we use 5 different datasets using various structural characteristics. For example, we have a small dataset like 62 E. coli, 7 humans that are very closely related but different genomes, 7 apes, that is related genomes from the same superfamily but from different species, and so also like 11 huge conifer plants and 100 humans. So for the individual results, now this is a very busy table, and actually this is a subset of the table that appears in the manuscript. So let's not dig into too much details into this table, and let's try to parse 1 or 2 rows. Say for this human data set and this 16 threads row, if we use k equals 31, then Bifrost builds the de Bruijn graph in like 1 hour and a half using 27 gigs of memory. DGSM takes like 18 minutes using 38 gigs of memory. Tupacco takes 6 minutes and a half using around 5.5 gigs, and Cuttlefish builds the graph in 3 minutes and a half and using 2.8 gigs of memory. So if you see the table and the timing performance, there are like 14 experiments here. In almost all the experiments, the timing performance Cuttlefish is the best among the tools, except just one case for this SugarPine genome and Keiko61. And the memory performance is actually the best one in all the experiments. That is, in all 14 settings, the memory usage is the best one among these 4 tools. To assess performance in for colored de Bruijn graph compactions, similar to the earlier results, here the timings are in minutes and memory usage is in gigabytes. So, like the earlier results, we are performing the best. For example, for this 7 humans dataset, Cuttlefish takes like 21 minutes and uses 2.88 gigs, and it's 2.9 gigs of memory, whereas Patro takes an hour and 6 gigs. DGSM takes 30 minutes and 38 gigs, and so on. There are some blank rows here because we didn't perform these experiments for Bifrost and DGSM in these 2 datasets. Extrapolating that, our hardware would not have enough memory to run these experiments for Bifrost and DGSM. And sadly, Patro had a fake fault for this 100 humans dataset. But I mean, the points to be noted here is that Cuttlefish performs the best in terms of time and memory, even for like colored compacted de Bruijn graph compaction and individual compaction. All the steps in our algorithm, that is all the 4 steps, the K-mer set construction, the hash function construction, and the actual 2 steps of graph traversal are very scalable in terms of timing, in terms of threads. I mean, actually this plot is shows a detailed, shows much more information. The first step, that is enumerating the K-mers, it saturates at around 10 threads. The next step of hashing construction, that is using the BB hash algorithm but heavily modified for our needs, it saturates at around 24 threads, and the last 2 steps of that relate to actually traversing the de Bruijn graph scale almost linearly up to 32 threads. So to wrap up the talk, we introduce this new algorithm to compact colored de Bruijn graphs from sets of references that shows promising results. The improvements stem from this novel modeling scheme of de Bruijn graph vertices with using a DFA. The tool is publicly available and implemented using C++14 and available at this URL. Thanks for attending the talk. So if you have any questions, any queries, any confusions, I'll be happy to answer.
Thank you very much. Really appreciate it. So we have one question already, and is it possible to use Cuttlefish as a C++ API or a header library?
We do not provide like a direct library that can be used. I mean, this can be done using just a few modifications, but at this point we don't have it, but we can provide it in future. It's possible.
Okay, thanks. Do you think that further improvements on runtime are possible?
Yeah, that's a great question. I mean, if we see this speed up plot here, the K-mer set construction, that is the first step, it saturates at around 8 threads. So maybe we can, if we have a better K-mer enumeration tool or algorithm, the total runtime will decrease. But the actual graph traversal steps, these are quite fast already. But the first 2 steps, that is building the hash function using the K-mer counter and the MPH algorithm, if these 2 can be improved, then yeah, I think the runtimes can be lowered further.
Okay. Well, nice work on Cuttlefish. Then, have you tried K-mer tricks to enumerate K-mers?
No, not yet. We are using the KMC3 algorithm, but we'll definitely look into K-mer tricks. Thanks for the suggestion.